Hi, everybody. I'm going to go ahead and get started. Hi, so the Center for Integrative Environmental Health Sciences and the Department of Pharmacology and Toxicology welcomes you to our Environmental Health Science Series seminar. Our speaker today is Dr. Timothy O'Toole. Now, Dr. O'Toole received his Ph.D. from the University of Michigan in biological chemistry, where he studied defects in purine metabolism. And he's held prior positions at the Scripps Institute in La Jolla and also at the Cleveland Clinic, where he studied integrin signaling and platelet and endothelial biology. Today, he's an associate professor in the Department of Medicine at the Environment Institute. He also holds associate appointments in the Department of Biochemistry and in the Department of Pharmacology and Toxicology, and is the director of the Metabolism and Toxicity Core at the Louisville Superfund Research Center. His current research efforts focus on the effects of environmental exposures, specifically particulate matter, air pollution, and volatile organics on cardiovascular outcomes. So additionally, Dr. O'Toole serves as an editor for the journals Circulation Research and Toxicology and Applied Pharmacology. And today he's going to talk to us about microplastics and cardiovascular disease. So I'm going to hand it over to him.
All right, thank you very much. Okay, so today I'm going to give a brief talk about a recent pilot experiment that we've done.
Sorry, go ahead.
This pilot experiment that looked at the effects of microplastics exposure on health outcomes, and specifically cardiovascular outcomes. So the hope is that these data from this pilot experiment would generate some new directions for the lab. So this is a new area for us and a pilot experiment, I welcome any feedback, input to these studies. So I think there's a large body of epidemiological and experimental data that suggests that there are associations between environmental exposures and CVD risk or adverse outcomes. And I think we're all very familiar with the work here and at other places that have looked at the effects of particulate matter, air pollution, volatile organics, chemicals, and other metals that contribute to cardiovascular disease. In recent years, there's been an increasing awareness and concern about environmental plastics deposition. So a few facts. It's estimated that there are 360 million tons of plastics that are produced annually, and this number will increase threefold by 2050. A large of these plastics end up in landfills or aquatic systems, about 4.9 billion metric tons. Under these conditions, there's a gradual degradation of these particles, both due to the physical wear and tear, as well as to some physical hydrolysis, oxidation, and chemical processes, which break down these larger polymers into smaller secondary microplastics. In addition to these secondary microplastics, there are plastics that are manufactured in the size range for commercial purposes. These are the so-called primary microplastics. And as mentioned, these both secondary and primary microplastics have found their way into the water systems and food chains where human exposure is inevitable. So what are the major routes of human microplastics exposure? The primary one would be through ingestion of food substances. And indeed, in some fish samples, if consuming fish, again, depending on how much you consume, it's estimated that you can uptake or intake 40 to 50,000 particles per year. And this estimate is almost certainly an underestimate, as there's uncertain quantities or uncertain data regarding the microplastic content of meat, poultry, and vegetables. In addition to food consumption, the consumption of bottled beverages also induces a supply of microplastics. If one consumes mostly bottled water, you can consume up to 90,000 particles per year. Another recent study suggested that a human could typically eat 5 grams of plastics per week, which is equivalent to a typical credit card. The second route of exposure is through inhalation. Microplastics are of low weight and density, and they can easily become airborne. And it's estimated that you could inhale 40 to 70,000 particles per year. What goes up must come down, and there's increasing awareness that some of these airborne microplastics can fall back to the earth. And indeed, there's an idea that's come about that plastic rain is a new acid rain. And in a recent study, it's been found in certain areas of the United States, mostly in the national parks of the western part of the United States, that over 1,000 metric tons fell, which is equivalent to 120 million plastic water bottles. Final way whereby humans can be exposed, although probably minor, is by absorption through the skin. So what is the fate of these ingested microparticles? Or microplastics rather. A lot of the early data has come from mouse studies, and it's been identified that they can accumulate in several tissues, including liver, kidney, spleen, and the gut. There's also evidence that they can be translocated into circulation. They can cross placental barriers, as well as the gastrointestinal mucosa. And depending upon the type of diet you eat, you can absorb more or less of these. In humans, there's been some limited studies, but nevertheless, microplastics have been found in lungs, feces, urine, and breast milk. In many of these outcomes, the distribution of these microplastics into tissues are dependent upon the size of the microplastics, its hydrophobicity, and its charge. A second concept that has been developed is that microplastics, although they may have inherent toxicity themselves, they may act as efficient carriers for other toxins. That is, when they are in the environment, they can absorb metals, persistent organic pollutants, or other chemicals, as well as microbes. And given that they can be distributed to various tissues in organisms, it's an effective way to concentrate these toxins in certain organs. So what are some of the biological consequences of microplastic exposure? Well, again, these are studies that are pretty much in their infancy, but some of the limited data to date suggests that they can induce inflammation, increase oxidative stress, induce changes in the gut microbiome, and alter the cellular bioenergetics. As some of these outcomes are some of the underlying factors that contribute to cardiovascular disease, we are interested in asking the question, does microparticle ingestion also promote CVD risk? So to do this, we designed the following study where we gave mice normal water or water containing polystyrene beads. We used 5 groups. One group was our control, which had normal water. And then we used our microplastics, our polystyrene water, which consisted of 2 different sizes, both 5 microns and 0.5 microns, and 2 different doses of each size, 0.1 microgram per mil and 1 microgram per mil. We kept these mice on their drinking waters for 12 weeks. During this time, we could record certain outcomes such as glucose, fasting plasma glucose, body weight, and metabolic activity. And then at termination after week 12, we could collect blood and other organs and measure several different parameters as listed over on the right-hand side. So here are some of our initial observations. The first was body weight. So illustrated here are the changes in body weight from baseline, so prior to supplying with the different drinking water supplies. As early as 3 weeks, we saw that there were 3 different groups had a significant increase in body weight compared to our control in the first lane. And although there was some variation throughout the remaining time courses, at both 9 weeks and 13 weeks, there were 2 groups which had significant weight changes compared to the control mice drinking normal water. At 9 weeks, after 9 weeks of analysis, we did a DEXA scan analysis to identify or to determine body composition and percent fat. We saw that 3 of these groups, exposure groups, had increased fat content compared to our control. Another endpoint we looked at was glucose homeostasis. First, as measured by fasting plasma glucose levels. Again, as early as 3 weeks, we saw that 3 groups had increased levels of fasting plasma glucose compared to our control. Again, there was some variation over the remaining time course, and the glucose levels actually fell over the 12-week time course. But at the termination, we saw that this group receiving the 0.5 microns at the high dose, 1 microgram per mil, still had higher levels of fasting plasma glucose compared to our control. To gain further insight into glucose handling, we performed a GTT. This was done between 9 and 10 weeks of exposure. However, there was no difference between the groups in the area under the curve. We also performed at termination using this group, which demonstrated the highest glucose levels. We performed an ITT, insulin tolerance test, versus control. But again, there was no difference in the ITT area above the curve between the groups. Collecting blood at termination, we measured insulin levels from all the groups. We saw that, again, this group, receiving the 1 microgram per ml dose of the 0.5 microgram beads had increased levels of fasting plasma glucose, and consistently it had higher HOMA-IR scores. Finally, with regards to the kind of metabolic measurements, we looked at plasma lipids. Again, these were obtained at termination after the 12 weeks of consumption. There were no differences in total cholesterol, LDL, or triglycerides, but the final group did show a statistically significant increase in HDL levels. So the major observations were that some groups had increased body weight, which was consistently increased percent of fat. They were increasing in adiposity. And one group, especially this last group, had increased HOMA-IR scores suggestive of insulin resistance. Given these phenotypes, we put these mice in metabolic cages to measure several different parameters of intake, respiration, and activity. However, when we did this analysis, we really saw no difference in any of these measures between the control mice and exposed mice. That is the mice, again, this one group, which showed our most abundant increase in insulin and glucose. From prior studies from our group and as well as others, we've shown that the inhalation of air pollution particles impacts or restricts the growth of some hematopoietic stem cell populations, suggesting that there may be larger long-term differences in circulating blood cell populations. So we were interested in seeing if the same thing was going on in our microplastic exposures. So at termination, we collected blood and did a complete blood cell count. We saw that there were no differences in white blood cells, lymphocytes, neutrophils, or red blood cells. However, we saw an interesting increase in eosinophils and basophils, but this increase was apparent only in the mice which were consuming the 5 micron beads, but not the 0.5 micron beads. There was also a minor increase in monocytes in one of these populations, and a trending decrease, but not significant, of platelets in several groups. We also did a couple of other measures that we typically do by flow cytometry. One is to look at the extent of platelet aggregates. This is a measure of platelet activation and thrombogenicity, looking at double positive events for CD41 and CD45. However, we found no differences between the groups. We also, in our mouse experiments and air pollution exposures, typically look at circulating levels of EPCs. These are the cells that are important in vascular repair, defined as FLICSCA positive cells. However, again, here we saw that there was no significant change between the groups. So again, the major findings so far were that we had increase in adipogenesis in some of our groups, as well as some indication of insulin resistance. So next, we were interested in trying to identify what might be the mechanisms of this increased adipogenesis. And the first kind of question we had was whether there were changes in the gut microbiota induced by microplastics consumption. And this is because there's evidence, well-published evidence, that obese humans and mice have a lesser diversity and richness in the bacterial component of the gut. And consistently, it's been shown that the consumption of a high-fat Western diet, which leads to obesity, is also associated with decreased bacterial diversity. And the second kind of published finding is that, at least at the bacterial genus level, that there is an increase in the relative abundance of the firmicutes over the bacteroidetes in obese humans and mice. And so why is this? There are several explanations as to why the identity and composition of the gut microbiome might lead to obesity. First is that the presence of these species might enable the fermentation of polysaccharides that are normally not digested by humans. Secondly, these bacteria might increase the production of short-chain fatty acids. They might lead to decreases in fatty acid oxidation and increase inflammatory responses. So there's several possibilities whereby the nature of the microbiome might increase or lead to adiposity. So in our studies, we collected feces after 12 weeks of consumption, again, using the feces from the control mice, as well as this population which had the most changes. And we did our 16S RNA sequencing. And what we saw here, looking at the diversity, both on the left-hand side in a PCA, principal component analysis, as well as in box plots on the right-hand side, that those mice consuming the polystyrene containing beads had a decreased diversity compared to our control mice. And again, this is consistent with the literature that suggests that decreased diversity is associated with obesity. We also looked at the bacterial genus of our gut microbiota. We saw an increase in the firmicutes over the bacteroidetes in our polystyrene consuming mice, again, consistent with literature reports. In addition, in the more lower, these were the two most abundant genus of the bacteria. There are some relatively minor species, but again, when looking at these minor genus species, we saw that there were changes in the polystyrene consuming mice versus the control. Now we've seen that there is increased adiposity, and increased adiposity and inflammation is often associated with cardiovascular disease. And that's because dysfunctional adipose secretes inflammatory mediators, adipokines, and thereby contributing to large-scale cardiovascular disease such as atherosclerosis. In particular, there's abundant literature suggesting that perivascular adipose tissue, that adipose tissue surrounding the vessels, that dysfunction of this tissue is associated with increased atherogenesis. Again, both by secreting inflammatory mediators and signaling various intermediate signaling pathways, leading to inflammation of the tissue and the increased adhesion and of immune cells and the initiation of atherosclerotic plaques. So to see if there were any changes going on in PVAT from our exposed mice, we isolated this tissue, again, from these same two groups. We did the RNA isolation, cDNA synthesis, and screened an adipogenesis gene array of 84 adipogenesis-related genes. So from this analysis, we found that there were 14 genes that were decreased by greater than 1.5-fold, as well as 5 that were increased by greater than 1.5-fold. So from this analysis, a couple themes came out. One was that there was an inhibition or decrease of Wnt signaling. Wnt, which is wingless, and int1, which I think derives from the Drosophila homolog. These proteins are a family of secreted proteins which bind to GPCRs and signal in an autocrine and paracrine fashion. They are typically involved in cell fate determination and embryogenesis, as well as some regeneration and proliferation and migration events. Specifically, in mesenchymal stem cells, which can differentiate into myoblasts, adipocytes, or osteocytes, in the presence of Wnt signaling, there's a preferential differentiation of this tissue into myoblasts and osteoblasts. In contrast, Wnt seems to inhibit the differentiation of pre-adipocytes into mature adipocytes. This is a schematic of the typical canonical Wnt signaling. In the absence of Wnt signaling, there is a 5-protein destruction complex, which phosphorylates beta-catenin, its ubiquitination, and its degradation. And in this case, beta-catenin cannot bind to its targets in the nucleus and promote the transcription or promote transcription factor expression. This leads to adipocyte differentiation. In contrast, when there is Wnt signaling, it binds to its receptor, the LR6 fizzled complex. It recruits this destruction complex to the cell membrane. Beta-catenin is now intact. It is able to go to the nucleus and induce transcription factor expression. And in this scenario, there is a inhibition of adipocyte differentiation. So some of the gene expression changes we saw fit in nicely into this canonical Wnt signaling pathway. First of all, we saw a downregulation. The expression is 0.4-fold of one of the ligands, Wnt1, of this signaling pathway itself, again leading to adipocyte differentiation. The most upregulated gene in our expression array was DKOF1 or DKK1, which is upregulated by a 4.2-fold. This is a natural endogenous inhibitor of Wnt signaling. It binds to the LRP and LRP6 and fizzled receptor, inhibiting Wnt binding. So again, with the inhibition of Wnt signaling, you get adipocyte differentiation. And finally, we saw that GATA3, a transcription factor which inhibits adipocyte differentiation, this was downregulated. Its full expression is 0.5-fold or 0.05-fold. So by inhibiting this inhibitor, you would stimulate adipocyte differentiation. So the gene expression changes we saw were consistent with the phenotype that we observed. Second major theme from our gene array analysis was that there might be a decrease in adipose browning. So I kind of have a rudimentary understanding of adipose, but there seems to be two major types, the white adipose and the brown adipose. And there are also intermediate types such as a beige type as well as PVAT. The white adipose has low mitochondrial content, but very high levels of absorbed lipids. In contrast, the brown adipose has more mitochondrial. It is more metabolically active, and that's because it's involved in thermogenesis. There also seem to be mechanisms whereby one type of fat can be converted into the other type of fat. Typically, this occurs in response to cold temperatures or exercise, where white adipose tissue can be converted into brown adipose tissue, both by increasing mitochondrial content and their metabolic activity. One of the molecules that increases the browning of white adipose tissue is BMP7, bone morphogenic protein 7. We saw in our array analysis that this was decreased. So it would suggest that with decreased expression of this, we'd have more white adipose tissue. And a second finding from our gene array was NRF1 was downregulated by 0.51-fold, and NRF-KO knockout promotes the whitening of brown adipose tissue. And again, our results are consistent with this tendency to increase the adipogenesis. So I've mentioned, in previous slides, some of these genes that are involved in adipogenesis or in browning, but there are also some genes, primarily P27 and CRAB1. The knockout of these genes is also associated with hyperplasia or obesity, and consistently we saw that these were downregulated in our mice consuming the polystyrene beads. And finally, since not only is the increase in adipose tissue associated with cardiovascular disease, but also the inflammatory content. So we looked at some of the typical inflammatory molecules, TNF-alpha, MCP1, and IL-6 in our PVAT tissues. Although TNF-alpha was downregulated, MCP1 and IL-6 are both upregulated, again suggesting that, at least in our PVAT, that it was becoming inflammatory and dysfunctional and might lead to eventual cardiovascular disease. So that's about the extent of what we've analyzed so far. And to summarize, we've shown that some groups, especially the larger dose, the 1 microgram per ml dose of both polystyrene bead sizes, demonstrated an increased weight gain, and that was probably due to an increase in body fat. Some of the groups demonstrated hyperglycemia. This was as early as 3 weeks of consumption of these beads. And we saw some hyperinsulinemia at termination. We've shown that there are changes in the gut microbiome which are consistent with increased adiposity. And we've shown in the PVAT tissue that we collected that there were gene expression changes, again, consistent with increased adiposity and inflammation. And finally, we have this interesting observation that those mice consuming the larger size beads had an increase in circulating eosinophils and basophils. So that leaves a lot of unanswered questions and some of the things we are thinking about in future studies. These changes in the PVAT, do they occur in other fat tissues as well? PVAT is only a minor part of the total fat deposed in a typical animal. We've looked at only polystyrene beads. Do other plastic particles have, such as polyethylene, do they have similar effects? And indeed, it's really hard to come up with an ideal experimental paradigm, since what one is exposed to in the environment is really a mixture of different types of particles and sizes, and again, what they absorb from the environment. Are any of these effects gender dependent? And really, some of these effects, are they exacerbated upon certain susceptibility states? So we're thinking about looking at different ages of mice, whether they're on high-fat diet or not, disruption of circadian rhythms, to see if these can exacerbate any of the effects of microplastics or enhance the initiation of cardiovascular disease. Finally, what are the mechanisms of this increased hyperinsulinemia? And what is the significance of this increase in eosinophils and basophils, which are typically associated with allergic responses in asthma? So I think I'll stop there. I just wanted to acknowledge the people who did the work, Jing and Daniel. We had some advice and guidance from Dan Conklin. Greg Shirk, Monica, and Xiao Hong also were involved in some of the analysis. And also like to acknowledge the funding sources and the cores who helped to get these studies done. So I will stop there and take some questions. Yes.
Have you considered doing cardiography to look and see if there's any effect on coronary flow?
Yeah, I mean, that's another disease model we can look at. I mean, we've kind of focused on the atherosclerosis, but yeah, there are many other cardiovascular disease models we can look at.
Your question in the chat box, that would be great. We're having some audio issues here.
So I actually had a couple of questions. I was wondering, you said normal water, and so I was kind of wondering what you mean by normal water, because in the animal room, they use acidified water, so I thought that might alter. They use acidified water. The water in the animal room is acidified if you don't change what they use. So that may change some of the dynamics.
Yeah, it's the water that they supply.
Because it may, change that homeostasis, and also the water itself, even though it's normal and acidified, it also may have. So it's just something maybe look in the future that may be altering metal homeostasis, which may go over to some of the phenotypes you're seeing, possibly. It's a carrier. Interesting.
Given some of your results with the microbiome and the cardiovascular disease, I wondered if you've considered systemic inflammation in these animals? It can also
yeah.
have used in a whole bunch of other diseases.
Yeah, we haven't looked at this first experiment. I mean, we just saw that some of these mediators were up in the PVAC tissue, but, and there were no, like I said, major changes other than eosinophils and basophils, no major changes in immune cell populations. But yeah, those are some things we think about. This is the first experiment, kind of a broad array of multiple endpoints to see what's going on, and now we can be a little more targeted. Yeah.
So I think it's interesting that your different size beads are leading to different effects in the mice. Do you know much about differences between biodistribution based on the particle size?
We haven't looked at that. There have been some studies. And I'm not sure if they have shown, there's some studies, I think I mentioned it, went to different tissues. I don't know if they look carefully at size differences and differential distribution in vivo. But yeah, it's something to consider. Chris.
Yeah, so riffing off that a little bit, is there a way to tag the beads so you can find out where they're going?
Yeah, some say that these muscle tendons.
And so we're ingesting these, so it sounds like a great opportunity to interact with what's on the fifth floor here. And then absorption and changes in gut barrier.
But that may require another control, right? Because the mice are not only ingesting the beads, but the whatever the tag is, the fluorescent.
Yeah, well, if you put it on the beads.
Yeah. I mean, yeah. Yeah. So the dose that you guys were chose, was it based how big is it, or relative to the everyday or annual consumption?
Yeah, that's another kind of big issue in the field is what is the proper dose to give. We can kind of make, ballpark figures of how many particles this is going to be, and I think I mentioned some figures that earlier on that are some estimates of how many particles a human can consume. So if we do these kind of ballpark figures, this dose is kind of the high end of what one would expect. So it's kind of, maybe overdosing. So we're looking now at lower doses and earlier time points to see if we can see similar effects. A lot of this stuff was done at termination and 9 weeks. So.
At least it's not 100 or 1,000 foot.
No.
And I was going to mention also, these mice have been brought up in a plastic cage and also drinking water from a plastic bottle. So how controlled are they? I'm wondering, have they been exposed to, probably microplastics all their lives. So I wonder if there would be a better control than, I don't know. I'm just, wondering.
I mean, we're seeing differences when we give them normal water. Yeah.
Right. No, I understand that.
Yeah.
But if you have a better control, maybe you may see a bigger effect.
Yeah, it's kind of hard to control everything the mice do unless you keep them in kind of very sterile conditions from birth. Yeah, it's.
So, were there any adipokine changes? You talked about IL-6 and MCP1.
I didn't look at it.
Huh?
I didn't look at it.
Oh, you didn't look at it. Another
Some.
suggestion for the lipid part is because mice, the C57s, mostly have HDL. That's why you are seeing changes in HDL, because 80% or more, up to 90% cholesterol could be just HDL. So you might want to do lipoprotein separation by FPLC and then look for LDL or VLDL. Then you might see if there are subtle changes you may miss, because it will get masked by high HDL.
Other question?
Yeah, I just have a quick- Did you look at water and food intake in the mice?
Yeah.
And did the plastics change the amount of water they took in or increase the amount of food they ate?
It decreased a little bit, but not significantly. I have that up. Yeah. the control mice drank 5 mils a day of exposed people, so it decreased a little, but not significant.
But that's for one day though, right? So,
This is a 1 day,
yeah,
24
so
hours.
over
is
time,
the
maybe,
metabolic
there
age.
may be some. Okay, well,
And
there's
a
a,
little bit, a
oh.
little less food to eat.
Yeah, because if you measure that over weeks, continuously monitor that there may be a difference. But
It would be interesting if they're eating less food but gaining more weight, like what's going on there. It's obviously been not a food-based thing, and B, as a result of whatever microplastics are doing.
right.
Yeah. Online questions?
Yeah, so Jenny Toyota is asking 2 questions. One, are the effects you see previously observed with microbiome disruption? Second question is, have you characterized the microplastics? Are they mixtures with absorbed contaminants? Do they stay suspended?
Yeah, so the second question, yeah, we get these from the company.
Right, so it's not.
We do, there she is.
Sorry.
Oh, wow.
I was saying the answer that you can do.
So, yeah, these are pristine feeds from the company. We changed, we went every day and educated more about it, so we try to eliminate any settling. We make a fresh water supply, I think it's every other day or every third day. And before we make this water supply, we do sanitation to break up any clumping that might occur. So we're doing what we can to make sure that there's single beads that they consume. Any other questions?
Well, it's from Jenny too. Any cellular uptake of microplastics done or planned?
Cellular
I guess,
uptake?
yeah, I guess, or in vitro or in vivo, I don't know what.
Yeah, not planned, but it's, something we could do. Again, we probably have to label them, wrestling or something, and then follow where they go. Other people have done these types of studies to see where they go. Yeah.
Yeah, I was also wondering if you've done any characterization of the microplastics after they've been in the water to see what may be absorbed. And the reason I asked if these effects were seen in other studies with this kind of disruption of the microbiome is if you could tell if these effects might be due to the microbiome disruption or directly from the microplastics or from the absorbed mixture that may be there.
Yeah, we don't have them analyzed. I guess it's taking the beads out of the drinking water or once they, yeah, we haven't done any kind of analysis. We're just interested mostly right now in seeing what kind of effects we can see. Yeah, that's something we can do.
All right, we have another question from the online audience. Idoya asks, great talk. Polystyrene B's exposure to cells in culture is known to cause mitochondrial membrane depolarization. Do you think it's related to the results you have observed in adipose tissue? I believe you were mentioning decrease in mitochondria, question mark.
Yeah, I think that's something we could do down the road. We haven't looked at it now. And it's just that we see, because of the decreased expression of those proteins involved in browning, which would be a more metabolically active fat tissue, it would suggest that we have an increase of whitening of the fat, which would be metabolically active.
I guess in vitro, the polystyrene is known to cause mitochondrial membrane depolarization. I think she's wondering whether you've looked at or consider that.
Yeah, well, that would have to be done like a single cell or tissue that we can harvest in single cell operations. Anything else?
What tissues did you take from the study?
PVAD, liver. I think that's good. Yeah.
Yeah, I thought PVAT was very specific because, so the adiposity, the increase in these mice, were they due to increase in PVAT? If that answer is yes, then I think it rationalized taking the PVAT. But was there, well, I guess we were trying to look at the association with the cardiovascular disease.
But I think the PVAT is a low percentage of the overall fat in the muscle.
Right.
So I would harvest other fat tissues, we'll do that next time. But it may be that other tissues as well, maybe have these gene expression changes to increase the difference.
Very stimulating.
Yeah.